Hello, thank you for looking at my poster. My name is Florian. I'm a postdoc in the Systems Biology and Data Analytics group at the Genome Institute of Singapore. In this poster flash talk, I tell you a little bit about a new method we developed for the clustering, the supervised clustering of single-cell RNA-seq data. So for anybody who performs single-cell analysis, your pipeline will look something like this. You start with your sequencing and preprocessing, including quality control, you go to clustering, annotation, and then you do the interpretation and downstream analysis. This slide already shows you that clustering is one of the first essential steps in this pipeline, and any error that occurs there will be carried forward to all of the other analysis steps, which really renders clustering to be a very important part which should be as accurate as possible. And because of this importance, many methods have been developed to address the clustering problem, and most of those methods try to solve clustering in an unsupervised fashion, which has the advantage that you can detect novel cell types, but it also comes with a few disadvantages. Some of those disadvantages are, for instance, that many different clustering methods tend to lead to different results. In other words, the clustering results are not very robust across different clustering methods. The second disadvantage is that batch effects have been shown to strongly influence the result of unsupervised clustering methods. While you can argue that methods have been proposed to adjust for batches, a major issue remains if your batch corresponds also to the biological signal you are interested in. For example, you analyze data at two different places in two different hospitals, and in both hospitals you had your control samples and your diseased samples. So the biological signal is one-to-one matching to the batch, which is the different hospital. And in these cases, it's very difficult to mathematically adjust for the batch. However, already back in 2017, our group proposed a method called reference component analysis, which is a supervised clustering method which can be a remedy for those mentioned issues. However, RCA, which is the short form for reference component analysis, had some issues in its first release. Its functionality was very limited, it wasn't really scalable, there was only one reference data set, and it was very difficult to integrate a method into existing workflows. So that's why we thought it's time for an upgrade, and we came up with RCA version 2, which we have extensively benchmarked for batch effect robustness. And this benchmarking is elaborated and explained in detail in a longer talk introducing RCA2, which is also part of ISMB, so please have a look at that talk as well. Furthermore, RCA is a very robust and fast clustering method. We have a comprehensive multi-species collection of reference data. We incorporated many downstream applications into the package, and it's now very easy to integrate RCA into existing workflows. So the core of RCA really is the projection step. Here we have a reference which is shown as a heat map on the left, where columns are cell types and rows are marker genes. And we have our query single cell set where the columns are single cells and rows are the genes. So in the projection, we compute the Pearson correlation between reference and query, which we call projection. This results in a new matrix where columns are still single cells, but rows are now cell types. And in an ideal world, as shown in the cartoon on the bottom right, we would hope that cells separate nicely by cell type in this new cell type space. So RCA comes with a predefined collection of human and mouse reference sets, which are based on bulk RNA-seq microarray or sorted single-cell RNA-seq data. Also, users can generate their own data sets using their own reference data. RCA2 offers the direct input of results from Cell Ranger, but also of pre-processed count matrices, for instance from Seurat, and those can then be subject to quality control and normalization. After projecting the data, it can be clustered using either hierarchical or more efficient graph-based clustering methods. With RCA2, We also allow the user to perform cluster-specific quality control, which is especially important if you have very heterogeneous datasets. After clustering, you can use RCA2 for several kinds of downstream analysis. You can look at heat maps that show you the projection result, you can look at U-maps, you can look at the cluster composition, and you can also compute the E-genes in both a one versus all and a pairwise manner, which you can use to obtain GO-term enrichment results or KEGG pathway enrichment. And compared to the previous release of RCA, we are much more scalable now. The projection time stays almost constant, even though the number of cells is increasing, as shown in figure A here on the slide. And also the memory allocated by the clustering is much less if we stick to graph-based clustering, which makes the method much more scalable for the future. So please try it out. The method is freely available on GitHub. We are working on a preprint, which is hopefully out very soon. And to stay up to date, please follow us on Twitter. And with this, I want to acknowledge my collaborator Bobby Ranjan, who helped a lot with this project, our other colleagues from GIS, from Duke NUS, and I have to thank ISCB for providing me a fellowship to attend the conference. I also thank you for your attention, and I hope you find the method very useful, and please ask any questions you like to ask.